In anatomy and physiology, or in general biology, the courses begin with just some basic chemistry because we are chemical. See all of this stuff? It's made of atoms and molecules and bonds. There's nothing here which isn't chemical. And so obviously, if I want to understand this stuff, then understanding atoms and molecules is important. Now, I have a number of videos on basic chemistry, and then I have a playlist on biochemistry, and that's what this video is going to quickly review now, to go through those molecules which are, essentially the stuff of living things. That's why they were named biomolecules or organic molecules. It was once thought that they were unique to living things, that sugars would only be found in living things, proteins, etc. We now know that's not entirely true. It is possible to form these organic molecules in the absence of life. They are even known in, say, meteorites from outer space. And so all you really need to do is to add a little bit of energy, and then things in the natural world, such as water, carbon dioxide, methane, etc., will form sugars and amino acids on their own. So while these aren't unique to living things, that's where the majority of them are. And certainly living things depend on these carbohydrates, these lipids, proteins, and nucleic acids. Now, it can be very intimidating to study these biomolecules because some of them are just simply huge. Proteins can be big and complex, like antibodies. And then, the nucleic acids, like DNA and RNA, I mean, they can form these huge chains. DNA molecules can form chromosomes, and chromosome 1 in human cells, if it was unwound, would be that long. All right, well, that's certainly very intimidating. How does one study molecules which are just so big? Luckily, it's maybe not quite as intimidating as you might think, simply because the big biomolecules, the big organic molecules, are often what we call polymers, which are chains of much simpler units, which we call monomers. And so if you want to understand the really long, complicated molecule, you can go a long way by understanding, say, the shorter, simpler molecule, and then understanding how they are joined. So for example, with carbohydrates. The simplest carbohydrates have, a handful of carbon atoms, so these have 6. These are simple sugars called monosaccharides. And as you can see here, they can be joined. They can be joined in what's called a dehydration reaction. It's called a dehydration reaction because an OH group is removed from one reacting molecule, hydrogen, an H from the other. And when you put those two together, you've removed a water molecule, an H2O. And the remaining reacting molecules are now looking for partners because they had been sharing electrons. They would like to share electrons again. And so they now join. And here we have, the beginnings of a more complex carbohydrate after joining those two subunits. And we can build, say, proteins in the same way. So if you were to take building blocks known as amino acids, here's the simplest amino acid, glycine. Most have something more complex than a hydrogen atom. Amino acids share the feature that they have an acid group, OH, and H. And each amino acid on one end and a free other. It's easy enough to do dehydration rates in which they are, and OH is removed from the other, uniting these two amino acids. Now again, we are on our way to constructing a complex carbohydrate. Of lipids, or different kinds of lipids. In this lipid here, that 3 separate acids are joined to of glycerol with 3 separate reactions. And so this type of reaction, this dehydration reaction, is a way that we can build more complex carbohydrates, lipids, nucleic acids. And so thus it's easy to go from simpler molecules to more complex. When we talk about the digestive system, then the opposite reaction is important, where we add water to a more complex molecule to break it into its simpler components. And so here we have sugars attached. We add an H1 and an O to the other. We can separate them into their two monosaccharide. As we digest the molecules, like smaller molecules that can be absorbed, we see a lot of reactions. We can do the proteins. So you can add one molecule into this, what's called a peptide here, and then separate the then into separate components. The same for this fat, but one molecule separate these all these fats. And so all of these organic molecules can be united and made more complex through dehydration reaction, or can then have down monomers using reaction. That being said, it would be appropriate to begin an overview of each of the four major classes of these biomolecules, starting with carbohydrates. All their individual atoms, geometric shape here. So many of them form ring-like structures, and you can see, kind of the carbons involved in the rings here. And this one single ring is referred to as a monosaccharide. If we were to unite two of these monosaccharides together, we would get a disaccharide. A short chain is known as an oligosaccharide, and then a long chain is known as a polysaccharide. And so carbohydrates can be broken into classes based on how many of these monosaccharide units one has in a chain. The most common monosaccharides have either 5 or 6 carbons in them. All of these 3 have 6, and all of these 3 that are what are called hexoses. So here are 3 common hexose monosaccharides in the human body. Glucose, which is C6H12O6, 6 carbons. It is a primary cellular fuel. So cells will get energy from glucose even when other sources like fats are present. So glucose makes these starch and polysaccharides present. Found in membrane receptors, but is a part of milk. And fructose is part of sugar, can be found on membrane. It can also be found singly because, for example, in the male reproductive system, accessory sex glands produce fructose as the energy source that a sperm will use while they are swimming. There is another type of monosaccharide which is important in the body that I'll get back to when we talk about DNA and RNA. These are referred to as pentose sugars because if you count the number of carbons, 1, 2, 3, 4, 5, there are 5 carbons in a pentose sugar as opposed to 6. So these are important as ribose and deoxyribose. Deoxyribose is missing an oxygen that you see here. So if you notice this blue oxygen in ribose, but not in deoxyribose. And it is this difference in the pentose sugar, ribose versus deoxyribose, which will make the difference between RNA nucleotides and RNA, and then DNA nucleotides and DNA. All right, and so I'll return to those pentose in that. So depending on which monosaccharides are joined, we can give disaccharides different names. So for example, if 2 glucose monomers are joined, then we would have 2 glucose glucose plus fructose to form the disaccharide sucrose, which is and then glucose and galactose, these form the lactose, which is sugar. Now, all of those had glucose in it, and then all of them had and all of them were disaccharides, but as we'll see in another couple of videos, they are so specific that in our digestive systems, the enzymes that break down one of them cannot break down the others, and so those differences are important. But once we're building these carbohydrates, we don't have to stop. We can just keep adding more and more monosaccharide units. So instead of stopping at 2, we could now add more to have a chain of, say, 5 or 12 or 17, etc. We could use glucose, galactose, and fructose, but there are also other ones as well. We need not go into, but there's a greater diversity of these sugars in your body. And we can make chains, especially on those proteins found on the cell membrane. So most and perhaps all of the proteins on our cell membranes are actually better called glycoproteins, because while they are part protein, they are also then part carbohydrate. They have a sugar aspect to them as well. And this is significant. So for example, if you have a specific protein on your red blood cells with this combination of sugars, then you are blood type O, if that is the only kind that you have. Whereas if in addition to this base, you then added a yellow the difference between blood type A and blood type is the precise blood type B. So we just want so these sugar chains that you see as sugars from your diet that are now part of the blood sugar, which then has glucose being absorbed into cells. That's to form protein attached, known as now this is their function theory. They're intended to please attach to citation. The problem with the pre-addition of hyperinsulinemia blood sugar is more of this is going. This is one of the reasons that if you were to ask why is I'm older, I don't realize it when I was younger. The answer is by that point in life you have more advanced glycation and more sugars have attached to proteins. This is why high blood sugar is a problem in the long term. These sugar groups can attach to LEN proteins in cloud vision. They can do a lot. And then also, glycation is an important health. You don't have to stop keeping going. As you can see, plants known as cellulose and other components which are carbohydrates. Lots and lots of carbons stuck to each other. Now, you might say, well, then that's great because I should eat paper and cotton and sawdust because all of this glucose would be a good source of food. We humans, we don't make the enzymes that break up cellulose, however, even though it's a polysaccharide. It can't be broken up by any enzymes that we make. I mean, termites do and cows do in the microbes they have in their guts, but we don't. And so therefore, this would pass through our digestive tract as fiber. And so cellulose is one of the most abundant organic molecules in the world. But even though it's pure sugar, it's just a long chain of glucose molecules. It's not something that we can digest. But there are two polysaccharides which we can work with. One is starch. That's a polysaccharide with lots of glucose monomers that plants make. And the other one is glycogen. That's kind of the animal version of starch. And as you'll study, perhaps later in your A&P experience, after a meal, you make insulin. And insulin then tells cells of your body, like the liver being the primary site, to then build a polysaccharide. So if you look here, these long chains of glucose monomers, that's what's called glycogen. And when we make the hormone insulin, our liver cells make glycogen and store sugar in the form of this polysaccharide glycogen. Now that's what happens after a meal. You're essentially saving your sugar for later. And so then when you're asleep at night and you need energy because you haven't eaten in a while, you make the hormone glucagon, which then tells you to break up this polysaccharide glycogen into the monomers glucose again. So about half of your day, you're in the absorptive state when you can be turning monosaccharides into polysaccharides, and the other half of your day, it's the reverse, and you can get to that later. When you talk about the digestive system, starting with starch, which can be half of the American diet, these long chains of glucose molecules, which you would then want to break into the individual monomers, the individual monosaccharides in the small intestine, right? And so starch, represented here, is a polysaccharide made of lots of glucose molecules that then, enzymes in our digestive system, like this amylase, would then have to break up, all right? So we have different polysaccharides which we interact with. The cellulose, which can't be digested and forms fiber. The starch, which is coming from plants and is a major part of our diet, given that it's most of bread and potato chips and French fries and rice and taco shells, etc. So we eat a lot of starch. And then the glycogen, which is how we store polysaccharides in our diet. And so, all of these will then be revisited when you talk about the digestive system. The second group of organic molecules is lipids. Now, lipids actually includes, diverse things, including from waxes to steroids to fats, oils, and others. And so there's diverse classes here. One of the things that these lipids share is their polarity. So here we have water and oil. And if you were to add two dyes, you'll notice first the oil and the water don't mix. But the blue dye mixes with water and the red dye mixes with oil. That's because the blue dye, methylene blue, is polar, and it mixes with water because water is polar. The red dye, Sudan dye, mixes with oil because the dye is nonpolar and oil is nonpolar. So while lipids do include diverse molecules, one of the things that lipids share in common is that they are nonpolar and therefore don't mix with water very well. A good starting place to talk about lipids would then be fatty acids. And so fatty acids are long chains of carbon and hydrogen. So here you can see a hydrocarbon chain, but it ends with an acid group, which will then determine the reactivity of the rest of this, which is rather inert. And so here's a fatty acid. But it turns out there are lots of different fatty acids. And that's why, say corn oil is different from sunflower oil, which is different from fish oil, which is different from chicken fat, which is different from beef fat, et cetera. The fatty acids can vary. There's lots of different kinds. They can vary in their length. All right, and so here you can see there are fatty acids. Some are short, some are medium length, some are long, some are very long. Another thing that can vary is the presence of double bonds. So these first two fatty acids, they only have single bonds connecting the neighboring carbon atoms. And so then each carbon can then form four partners. One carbon there, but then this one, in addition to binding to those two carbons, can bind to those two hydrogens. And so thus all of these carbons, they're full, and they have as much hydrogen as they can fit. In this chain, each one, this one can fit two, this one can fit two, et cetera. So they are saturated with hydrogen. However, atoms can also then form double bonds. And so here you can see carbons which are sharing 2 electrons each. That's a double bond. And that then means that this molecule is not saturated. It could fit 2 more hydrogens here, but because it's formed a double bond, then there's no electrons to spare. So this is an unsaturated fatty acid, okay? Now, you can have one double bond or you could have multiple double bonds and then be polyunsaturated. And so there are different types of fatty acids. They can vary in their length, whether they're saturated or no. They can vary in whether the hydrogens are on the same side of the double bond or not, forming cis fatty acids or trans fatty acids. And if you were really into health, there are health implications to cis fats versus trans fats. And then we have this 3-carbon molecule known as glycerol. And you can then take fatty acids and pick any 3 to join them to glycerol to form a triglyceride. So triglycerides form both. We pack them together. So for example, we're going to put these into a box. The fact that they're all straight means that you could pack them tightly into a box. It's easy to get them together. And in life, that would mean that they could become solid at room temperature. They would be a fat. So fats have straight chains made of saturated fatty acids. But this is not a saturated fatty acid. It's an unsaturated fatty acid. Notice that there's a double bond here, and that puts a kink in the chain. A polyunsaturated fatty acid would be even more so. And as you might imagine, if you had a bunch of those, it would be harder to pack them all together. And so then at room temperature, they would be less likely to be solid and more likely to be liquid. So this would be an oil. So the difference between fats and oils, while being both triglycerides, is the fatty acids that they have, whether they are saturated or unsaturated or unsaturated. So fats and oils, these together, these are the triglycerides. They are very important lipids, and as we study anatomy and physiology, they are energy storage molecules. They are important in our diet. They're energy storage molecules, as you can discuss later, because they're so very efficient. So if you had a pound of carbohydrate and a pound of fat, the pound of fat is storing twice as much energy as the pound of carbohydrate. So if your body has a choice, do I store energy in the form of triglyceride or as, say, a polysaccharide or a protein? Well, you choose the triglyceride, which is why we accumulate fat when we gain weight. We don't accumulate this energy as anything else just because fat is so efficient. There are other forms of lipids, however, in addition to the fats and the oils. So if one were to once again start off with glycerol, which is a 3-carbon molecule that can form 3 bonds, you could add 2 fatty acids to it. So there's one fatty acid, there's a second fatty acid. But then you could also add a third group, not a fatty acid, but rather a charged phosphate group. And this would then form what's called a phospholipid. This is also a kind of lipid. So it has glycerol, 2 fatty acids, but then a phosphate group attached to one of the carbons in glycerol. And as we'll see when we talk about the cell membrane, this is the primary lipid component in the cell membrane, and we'll go through its its characteristics in membranes later. So here's a cell membrane, and the phosphate group is polar. All right, so that will then face the outside of the cell. But then notice that the cell membrane has what's called a bilayer, 2 separate layers of phospholipids, one with the polar heads facing outwards, one with the polar heads facing inwards. So phospholipids, another important kind of lipid. At this point in my class, I usually then ask the class, I'm sorry if this is a personal question, is any of you on steroids? And feel free to not answer if you feel it's too personal. A lot of students look at me like, I've asked them, something which is, too invasive and too personal. If I say, are you on steroids? Do you have steroids in your blood right now? And so no one typically raises their hand, and I tell them, I want you to all rush to the hospital because you have a problem. And the reason I say that is because you should have steroids in your blood. You're in serious trouble if you don't. And that's because cholesterol is a steroid. A steroid, by definition, has these this pattern of carbon rings in it with various side groups. Cholesterol is essential for life. It's in every cell membrane. It's in all of the cell membranes of our bodies. If cholesterol wasn't in our cell membranes, then our cell membranes would be too fluid. So cholesterol is a steroid, and it is essential for life because, once again, it's a factor of cell membranes. But not only that, we can take cholesterol and change it into other things, like vitamin D, like aldosterone, a hormone, like cortisone, a hormone, or like estrogen. Estradiol is the major estrogen of the body. Testosterone is a steroid also derived from cholesterol. And so there is more to steroids than just, say, anabolic steroids or the anti-inflammatory steroids that one might take to minimize swelling. And this is very important. So for example, if we were to talk about, say, breast cancer and the role of estrogen in promoting cell growth, it would now become very important. All of the enzymes which start with cholesterol and in a series of reactions change cholesterol into testosterone, and then the final enzyme that takes testosterone and converts it into estrogen. The more of this which you promote in your body, the more cell division which you can have, and thus the higher the risk of breast cancer. And so steroids are very important. And as you can see, there's a lot of different steroids, and so their chemical differences very important to us. Here's an adrenal androgen that can be then converted into testosterone, and then once again testosterone can be converted into estradiol, the major estrogen. And so steroids are lipids. Waxes are lipids, the kind that might appear on fruits and vegetables, but which also line our auditory canal for protection from dust from insects. So earwax is a lipid in the human body. One final thing I also ask my students: has anyone ever heard of eicosanoids or the subclasses of eicosanoids, leukotrienes, and prostaglandins? And they typically say no. And then if I ask, has anyone ever taken a medication for your eicosanoids, being your prostaglandins and your leukotrienes? Very often they say no. And then I'll ask, no one's ever taken aspirin or Tylenol or Midol? And then it becomes clear that there's another kind of lipid which is very rarely spoken of, but is so incredibly important. So here are fatty acids. But it turns out that some fatty acids can function as signaling molecules, as hormones, and they are called eicosanoids. And we can break them into groups. So leukotrienes are used, say, by your immune system and very important as immune cells talk to each other. But here's just one example of why that's important. If you wanted to look at where the first double bond is, starting with the far end, not the end that has the carboxylic acid, you could start numbering them. And so notice here's one that starts at the 6th carbon, while here's a double bond. And notice that would start with the 3 carbon. And so therefore one could say, all right, so let's distinguish between them. Let's call one an omega-6 a fatty acid, and the other one's called an omega-3 fatty acid. It turns out that we have different sources of fatty acids in our diet. And if you were to say, eat fatty acids from, say, the beef and chickens, that you would have different fatty acids than if you were to eat fish. Some of these are essential fatty acids, so your body can't make them. You have to include them in them. You can include it in cell membranes. And these fatty acids which we ingest can then be converted into these local hormones. Almost every cell in the body is capable of making or responding to eicosanoids. So not only are they hormones, but they're very widespread hormones in the body. And some are pro-inflammatory and some are anti-inflammatory. Now, inflammation is normal, but excess amounts of inflammation can make your heart disease worse, can make your asthma worse, can make your predisposition to cancer worse. And so the fact that fatty acids that we eat can be converted into hormones, which then can send signals to the body, which can either make health conditions worse or better. This is, very this is certainly important. Some of these are essential things which have to be incorporated into the diet. And so eicosanoids are another class of fatty acid which can be used as hormones. So lipids are a diverse group of molecules, which would include triglycerides, the fats and the oils, which vary in whether they are saturated or unsaturated. The steroids, most abundant, but then also things derived from it, like estrogen, testosterone, cortisone, vitamin D, and others, waxes, and then icosanoids, which are these local hormones. Okay. Third group of, oh, you know what? I'll break this into two separate videos. So I'll stop here and then make a second half for the next two groups of biomolecules, proteins and nucleic acids.